evening, everyone. Editors, if you can, put some X Files theme song here.
Mhm.
Because we are doing a tale from the X Files of Psychology. Spatial location of response key selects a neutral system for visual recognition. Now, this may seem like a very confusing sentence, and I think at the start it's going to be, but I think at the end we're going to get a clear view of hopefully what Dr. Arnold is going to be presenting. And just a brief introduction to Dr. Arnold. Dr. Arnold is a cognitive psychology professor at Rutgers University. He had earned his PhD from Stanford, which, shout out because I'm from the Bay Area.
Mhm.
And so it's nice to get a little recognition from there. But he's been at Rutgers since 1975. His research and a lot of his work really focuses on human cognition and neural systems, and we'll find out what those are today as well, and how these two really large topics contribute to semantic memory and instructional technology. Dr. Glass is also a Fulbright Fellow. He's a member of the Association of Psychological Science, and he's the recipient of many honors, including the James McKinnon Kettle Fund Award. Dr. Glass, it's all on you.
Okay. Um, all right, so let me start by describing a very simple task, which is designed to be simple. This has many versions. This version of the immediate recognition task, person sees a string of, say, four consonants, and then they say a single consonant, and they have to respond as rapidly as possible by pressing one of two keys, whether the test consonant was in the study string. The idea here is that encoding is easy, so they're going to get perfect encoding. The task is immediate, so there's perfect retention. And what you're really interested in is what's the mental comparison process between the test item and the study sequence, and how long it's going to take you to decide whether or not the test item was in the study sequence. That is, response time is going to be a clue to the underlying sequence of computations that gave you the answer, and therefore this is going to be a window into how the brain actually works. Now, when you do the task, a good scientific method is half the time for half of your participants when the right key is assigned the same response. So they press the right key if the test consonant was in the study sequence. And for the other half of the participants, you make it the right key is assigned a different response, and the left key is respond the same response, the exact opposite, so that there's no effect over the whole experiment of which key is assigned which response. Response assignment, therefore, when I do the experiment, is included in the analysis because it is being systematically varied either between or within subjects. Now, when I did this experiment, I got what was a very surprising result, which is that the results were completely different in terms of response time, depending whether the right key was assigned the same response or the different response. If you look on the right-hand side, when the right key was assigned the different response, it was clear that what was happening was that the test constant was being compared to the study sequence from left to right. And if a match was found immediately, a same response was made. But if in fact no match was found and the entire sequence was examined, then a different response was made. That's the only possible interpretation of what the response times were like when the right key was assigned a different response. However, when the right key was assigned the same response, if you look on the left, it's clear that that's not happening at all. The test consonant is not being compared to the study sequence at all. In fact, the way responses are being made is if the test constant is perceived, this is a perceptual effect, not involving comparison with the what's in memory at all. If the test constant is perceived as recent, that is, someone perceives it as having just been seen before, then they infer that it was on the study sequence and they respond same. But if
Mhm.
the test constant is perceived as novel, that is, it seems like something new, then they infer that it was not just on the study sequence and they respond different. So we have two entirely different kinds of information and computational process that's being used depending just on which key is assigned the same response and the different response. Furthermore, consistent with this, this experiment was done when fMRI was recorded, which allows us to look at brain activity when people are performing the task. And when the right key was assigned a different response, and and you had the test constant compared from left to right with the study sequence, you get both hippocampus and right caudate activation. And in fact, we already know from other studies that this is not a shock because because the caudate nucleus of the basal ganglia has a number of functions. One function is it's always involved in motor control, and it's particularly involved in sequential motor control, but more generally in any cognitive activity, if you're ever dealing with a sequence of something that you're generating or comparing, you always get caudate activation. So the caudate, originally a motor part of the brain, is now an important cognitive structure, and whenever we keep track of a sequence of things, the caudate is activated. So whether you're talking about
Dr.
your,
Glass?
yes,
Sorry. So I just want to see if I kind of got in my brain of how this whole study went. So there's like a monitor in front of me. I have two keys, right? This is
right,
my right, this is my left.
right.
And then there's going to be a sequence of constants,
A
a
string
string, a
of
string,
four constants, yeah, that
four
appears,
constants
and
that
then
appears, and
yeah,
then immediately there's going to be a choice of a random constant,
yeah,
and you're going to see if that constant was in that string.
exactly,
And then so the right key is assigned for some subjects, this is the same, and then
yes,
left key is different. And
yeah,
then for the other subjects, it's this assigned opposite. The right is
yes,
different. Okay, so now we are looking at subjects that had the key that was the right key that was linked to the answer being different. So if I
yes,
saw a string of constants, and let's say it was like XMDR, and the next letter was M, one of
yeah.
my choices, and I knew that it was different, I would click the right key.
Yeah, but don't forget, this also includes the data if M was in the string and you press the left key. Um,
Mm-hmm.
this is an effect of response assignment and not response. Okay,
I
so
see.
you
I see.
get a different result for both keys when the right key is assigned a different response versus the left
I
thing.
see.
So
Yeah, yeah.
that's important. Okay,
So
so
we're looking at the actual response when there's a response assignment
yes.
that's different or novel to what we usually know.
Well, there are two ways you can have response assignment. You can either have right key same and left key different, or right
Mm-hmm.
key different and left key same. And it turns out that difference that seems so trivial that most experimenters don't include it in the analysis tends to have a huge effect in this task.
Yeah.
And first is the behavioral data makes it clear that two entirely different computational um, operations are being performed on different data literally in the brain depending on what your response assignment is, and then the fMRI reveals that these two different sets of computations are unsurprisingly now being performed by literally two different parts of the brain. If you look at the part of the brain that's activated when the right key is assigned a different response, it's an entirely different part of the brain that's
Wow.
activated when the right key is assigned the same response. So it
Yeah.
reveals that we have these two separate systems in the brain that can both perform this very simple task. And uh, look at the effective response assignment, you see the effective response assignment, this very simple task, selects one of these two systems, which ends up controlling your response to it. Right. So
Yeah.
all of this is surprising. The first thing is surprising is that there are these two completely different systems in the brain that are both available for this very simple task. And the second thing that's surprising is that just response assignment, which comes after the whole thing is over, that's actually going to determine which of these two neural systems is going to contain your response. Right? It
Yeah. If
determines
I
it.
was a subject, like for example, if I was a subject in in this study and I had gotten right key different, my response time would be so bad because my brain automatically goes, right is same, left is different. I don't know why. Maybe there's a
Look,
reason. We'll find
look,
out more, but
look, you think that's true, but when you'd actually be in the experiment, you'd be like everyone else. And you have to understand that
mm,
people are responding in between half a second and maybe three-quarters of a second. So it's just like you see it, you know the answer, and bang, you hit a button. You don't like have this great introspection into what you're doing. It's just such an easy task that you just see the test item and bang, you're hitting one button or the other. As I said, between half and three-quarters of a second. So everyone does very well on the task, right?
how are
And
you how
yes,
are
go
you
ahead.
able to sorry, how are you able to capture did you have um the subjects, like did you have them like hooked onto a like monitor that read the scans? Like how were you guys
Oh
able to
no, okay,
capture?
okay. Yeah, well, you normally you do the experiment just sitting in a chair in a lab, but when you do an fMRI experiment, they get
Uh,
slid into the big fMRI machine with a big magnet over them, and then they have the little response box, the non-magnetic response box in front of them. So they're doing it inside the fMRI machine, so
oh
that they,
wow.
yeah, so they can't be super claustrophobic if you're in the experiment.
Yeah,
Yeah,
you're
yeah.
like immediately out.
Yeah, yeah, yeah. Well, by your own choice, you see. But if you're not
Yeah.
claustrophobic, and once you roll into the fMRI machine, obviously you forget all about it, but you're just banging the keys and you come out here. But that's how you have to do it inside the
How
machine.
long does it usually take?
These sessions are about half hour, so it's not a
Okay.
burden on anyone. No. Yeah.
Okay, okay.
Yeah. All right. So we get two parts of the brain. For one condition, whenever there's sequential processing, you get activation of the caudate nucleus of the basal ganglia. And as I said, that's not a surprise because that's always involved in sequential processing. However, when you have the right key equals same, then there's no caudate activation at all, right? The caudate is not involved. That's just the hippocampus, which is a key structure involved in perception and particularly in perceptual recognition. And that's clearly the part of the brain which is generating the experience of recency or novelty, which you're using to make the decision. So we have very clear results. We have two different behavioral results correlated with two different parts of the brain, which you would think would be the parts of the brain that generated those different behavioral effects. Okay, so these are
Could
very
you
clean data. Yeah, go ahead.
could you remind me or and our viewers, what was caudate activation?
Okay, caudate. Okay, all right. In the brain, you have a structure. You have a larger structure called the basal ganglia, which was first identified as being essential for all voluntary motor control. So every time you're making a voluntary movement, the basal ganglia is largely in control. And within the basal ganglia, it has a central structure which looks like this like a fish with a long tail, and it's called the caudate nucleus. And so people don't say caudate nucleus; they just say caudate. And and that's the structure which always is active whenever you're doing anything motor or whenever you're doing anything sequential. So it's going to be active when you're moving your hands, but it's also going to be active when you're remembering or saying or punching in your phone number because that is, by definition, a sequential structure, and
Yeah.
anytime it's sequential, the caudate nucleus is the one that encodes sequences of all kinds.
Is that how like a lot of musicians will have like muscle memory because there's like a sequence to like music, for example, right? Kind of bringing art in
No,
here,
no, no,
but
that's very, look, that's very insightful. Okay, that's right. What people call muscle memory has nothing to do with the muscles, but it is a real thing, and it absolutely would involve the caudate nucleus. Okay,
Oh,
it would
okay.
involve why you can just by doing this, it all comes out right, is because the caudate is doing. I mean, in fact, we know these kinds of things now. In that, for example, if you famous study, you looked at the violinists and the um, and you have different hemispheres. You have the right hemisphere controlling the left hand, and the left hand controlling the left hemisphere controlling the right hand. So if you look at the hemisphere that's controlling the fingers of a skilled violinist, I mean a grandmaster, um, the fingers that are plucking the strings when they're playing, that part of the brain grows to 5 times the size as the part of the brain that controls the fingers that simply move the bow back and forth. Okay, so
Oh,
we can
yeah.
see at this level of the detail now how if you're going to have a very fine motor task, as you've talked about in terms of music, you see that part of the brain get bigger as you practice it more and more, and that's why you're so good at it. Generally,
Okay.
the size of the brain that controls the structure doesn't have anything to do with the size of the structure in your body, but how fine the motor movements are that you have to perform. So you have a
Mm,
whole lot of area of the brain involved with the hands, and a very small part of the brain involved with the torso. Because there isn't that much you do with the torso compared
yeah.
to any look what you do with the hands. Yeah.
Okay, well, next time I'm practicing my guitar, I'm gonna tell my friends, I'm gonna be like, I'm getting my, what was it, caudate nucleus. I'm
Yeah.
using up
You're
my
building.
caudate
You
nucleus.
look.
I'm building.
Yes, it's important to know that we all have designer brains that we've designed ourselves by our
Mm,
choices. Because when
cool.
you learn something new, you are creating permanent new cells in the brain. And therefore, over time, for the skills you develop, there's a larger part of the brain that's devoted to that than would be if you didn't devote that skill. Yes, so you're having a definite, real effect
Okay.
on yourself.
I just gained 2 lessons here. Work on my fine motor skills
Yes.
and learn new things every day. Great.
Yes, absolutely. Definitely learn new things every day. That's what makes your brain. I mean, it's often you read these health magazines which say if you exercise and eat right, get plenty of sleep, you'll grow your brain. But that's not right. If you exercise and eat right and get plenty of sleep, you'll generate a lot of new neurons every day. But if you don't learn anything new, all those neurons will die within 2 weeks. The only way
Mm,
you make those neurons permanent is by actually learning something new. And when you're learning something new, then they become permanent because they become part of the representation of the new things you learn. So people who learn a lot of things, they have much larger particular areas of the brain associated with what interests them and what they spend their time doing.
that's so cool. Brains are awesome.
They absolutely are, no question. Yeah, yeah. Okay, so we got this now, right?
Hmm.
Now, obviously, so this obviously has some implications. It's that response assignments, as I said, selects one of two different neural systems that can both perform immediate visual recognition. Um, Now, I thought that the discovery of the effective response assignment would elicit an enthusiastic interest from my colleagues who do this kind of work around the world, and that's because it was similar to something else that had generated a lot of interest. The effective response assignment on visual recognition is reminiscent of the well-known classic finding that the right and left hemispheres process visual and verbal stimuli independently in split brain individuals. Now, this is actually a very kind of artificial effect, which you can only which you're only able to do with a handful of humans in the world for a brief period of time before they developed better drugs, and they no longer had the problem that required you to cut their corpus callosum in half. But the fact that you could show the right and the left hemispheres separately doing things somehow elicited such huge interest that the work became world famous. The lead author won the Nobel Prize, and it entered into affected people's understanding of things. Like the expressions right brain and left brain come from this paper, and everyone knows what a right brain person is, left brain person is. Now, in fact, nothing in the original paper gives you any reason to believe there are such things as right brain people or left brain people, but it just shows you the influence of the paper that everyone in the world now thinks along the line. So this was very influential. Now, in fact, while that had very limited applications, now what I had is I had a methodology that you could do with anyone, not just a handful of individuals in the world who are damaged, and I could show two completely different parts of the brain could be isolated, and you could see what one part of the brain, and you could see what the other part of the brain did. So this, since you could do it with anyone, and you could do a lot more experiments adapting my basic methodology, we really had the opportunity to learn a lot more about how different systems of the brain particularly function, because we now could isolate one system or the other and precisely see what it was doing. So I thought my colleagues would find this very interesting, and they'd publish it, and people would talk about it. And I had completely misread my audience about what was going to happen. Okay, when I submitted, okay, what I should tell you is that psychologists, all scientists operate under the peer review system. The way the peer review system works, I don't know if your whole audience knows this, is that you send your report to a journal. The editor looks at it, and he sends it to reviewers who are other scientists, and they write back saying whether or not it should be published, and if it survives peer review, that's published, and if they object to it being published because they see flaws in it, then it doesn't get published. So I sent it to a leading journal called Neuron, and I wasn't really surprised that it got rejected there, because they reject almost everything they get because they can get away with it. I was more surprised when another journal, Learning and Memory, did it, and also Learning and Memory, it was surprising because 99% of the time, if you're an established scientist and you send it to the editor barely looks at it. He sends it out, or she sends it out to reviewers. For an editor to look at your paper, read the title and the abstract, and just send it back to you and say, we don't publish things like that. That's extremely unusual. Okay, so I already knew it was unusual that they didn't even think it was worthwhile sending it out for review. They just didn't think it was worth publishing at all. So now I send it to another highly prestigious journal called Neuroimage, and I knew that they had to take me seriously because I was one of the people that when other people sent reports in, they sent it to me for my opinion, whether or not it should be published. So they couldn't say I was a jerk because they used me to decide on other people's work. So what the editor did is he sent it back to me. He said, okay, I see this could be important, but what I need you to do is run 6 more subjects to have the data really good. And also, I want you to do these very specific analyses, which would make it very clear what the effects are. And also, when you send it back with all of the raw data that you collected, just in case when the reviewers wants to check it all out. So I did all that. That took a while to do.
Yeah.
But when I finished all that, the results were exactly the same. If you've looked at the original graphs and the new graphs, they look the same. So I sent it back to Neuroimage, and now what happened is rather surprising. This time, the resubmission was again rejected without review. But the reason why is that the editor said is that no one in the whole world of all the neuroscientists could be found who was willing to review my report. Right now, this is a truly astounding result. I happen to
Yeah.
enjoy reading about history of science, and as far as I can tell, the last time this ever happened to someone, that person was Galileo. And Galileo was the first modern scientist. So you can't go back farther than Galileo. Okay. What happened to Galileo was that after he perfected his telescope and he looked at the moon, he wrote a report with figures he drew. He was actually a world class Renaissance artist. Um, and said, look, the moon is not an absolutely perfect body. It actually is like the Earth, has mountains and valleys in it, and this is what the moon looks like through a telescope. And he got feedback, pushback from his colleagues in natural philosophy who said, that can't be right because Aristotle said it's a perfect body. And Galileo responded to them, well, why don't you just come to my lab and look through my telescope and you can see for yourself. And the response he got from his colleagues is they refused to look through the telescope. And this is not an apocryphal story. The correspondence still exists, documenting that's what his friend said. Oh no, I'm not going to look through the telescope to see what it looks like. And I know of no other case in history of this happening until now. This had happened to me. You see, I had this very important result, and the way my colleagues around the world, just rather than being bothered by something new, we're just going to refuse to review it, and therefore there will be no way to get published, and that's why it's truly an example of therefore belonging the X Files of psychology. I can't see how it would qualify better than no one wanting to admit it even exists. So now I knew I was in trouble, and I was not surprised when all the leading journals simply rejected it without review, without sending out review. This was too troublesome to let the world know about. But then I knew I had something that would help me, which was that my initial finding was not serendipity. Right? I actually had reason to believe that these two systems in the brain already existed, and I was deliberately doing that experiment to try and find evidence proving that the two systems that I had shown actually existed. And previously, another journal, the leading journal for perception, attention, perception, and psychophysics, had a special issue, and they asked for theoretical papers, and they had accepted and published for me a paper on the dual system model, which is the theory that these two different recognition systems exist in the brain. So in this earlier theoretical paper, which they had already published, I had already predicted the result I now found, proving that the two systems did exist. Essentially, my experimental report had confirmed this earlier prediction, this theoretical paper. So when I sent the article to Attention, Perception, and Psychophysics, I said to him in cover letter, look, you've already thought this was reasonable enough that you published a theoretical paper predicting this result. Now here's an experimental report confirming that prediction. So how can you not now publish
Yeah.
the fact that the prediction's been confirmed? So they agreed they were sufficiently backed into a corner that they did send it out for review. Now, when it came back, it was initially rejected, but it actually had gotten a favorable review. So I wrote an email back strongly objecting to the fact that they were rejecting it, and then the editor gave it another objection that the real reason why is he thought it could be explained by this other effect called the Simon effect. We don't have to go into this, but basically, after a lot of back and forth, two additional experiments were added that showed that the objections that the editor had were not valid at all. And eventually the report was finally published. Okay, so it's now part of the scientific literature that, in fact, response assignment can select different neural systems. Though that doesn't necessarily mean, given the initial reaction, anyone's ever going to admit that report exists or cite it or do anything with it. Okay? In fact, the entire review process had taken 4.5 years from initial submission until publication. And I want to point out that journals typically strive to publish things in 30 days, okay? So you can see how much dragging of feet, is involved when you're really trying to get something new done, all right? And unless you're like me and you're willing to keep fighting, it just never happens. Most people give up somewhere along the line.
That kind of drives me back to to kind of ask this question. Like I have some ideas as to why I believe this finding is important, or especially this like theory of dual systems and dual systems of recognition in our brain, how we sequence or how we recognize patterns. Um, why is it important? Like I have my own ideas. I'll talk about them too, but I want to kind of hear from your perspective. Like why should we know about this?
All right, okay, that is a very good question. I was at one point I was going to save the full implications to the end of the talk, but basically, right now we know remarkably little about how the brain is organized, and if my theory is correct, then what everyone else in the world believes now is incorrect. And if what they're operating under is an incorrect theory of how the brain is organized, they're going to fail at a lot of practical attempts in neurology, for example, to help patients who have neurologic problems. For example, there's been no progress in the last 60 years in treating dementia, such as Alzheimer's dementia.
Yeah.
And if you believe my work, that's because for the last 60 years, all the efforts to treat it have been directed to a part of the brain which is not the part of the brain involved in memory, therefore not the part of the brain which is
Mm,
actually being damaged and causing the dementia in the first place. And so they're never going to succeed. They haven't succeeded in 60 years, they won't succeed in 600 years if they absolutely are fixed on the idea that this is the only part of the brain that should be examined. So in terms of making progress and helping the world, if you don't have a correct theory to begin with, you never ever are going to get anywhere with your more
yeah.
practical goals. That's why people in general should care about
That makes a
this. Yeah.
total sense. For me, it also came up to like, like different communication styles and how to like quickly understand, oh, this person's not just like being aggressive because I have a past, occurrence with someone that's used the same language. Maybe this person's just, you understand that maybe they're, recognizing my communication style as something else, and maybe that's going back and forth. Like I was just kind of thinking about like communication styles and how even that is a pattern recognition in itself, right? Like body language included.
Yeah, all that's true, of course, and that's because, I mean, when people started working, they did it exactly the correct way. You start with the simplest model you have, and either it fits the data or it doesn't, but the way it fails tells you how you have to change it and replace it with usually a more complicated model. However, what's ended up happening is that, as you mentioned, there's lots of behavioral data that things are more complicated than people doing neuroscience today assume. Okay, their
Yeah.
models are just too simple, um, but they really, really resist the the implication that it's more complicated than what they're working on. It's going to be a lot harder to work it through, and so they're not happy with my kind of data, which if they accept it, means that it's demonstrably more complicated, and they need to do more complicated experiments and do more need more complicated theories because that would make the task measurably more difficult figuring out what's going on. So yeah, so that's the issue. I mean, if you're committed to a simple theory, then you just want to pretend everything I'm doing just doesn't exist.
Yeah.
Excuse me.
You're okay.
Yeah. So anyhow, despite all this, I mean, I undeterred, I keep performing experiments. Okay. And I slightly changed the task in a couple of ways. Now, instead of presenting a string, now I am presenting a sequence. So people see a sequence of digits, and then they see a test digit, and again, they have to respond as rapidly as they can. Whether or not the test digit is in the study sequence. Now, the reason why this particular task was selected was that besides generalizing my first result, if you go back to the origin of the cognitive revolution, where behavioral psychology was replaced by cognitive psychology, the revolution took place in the year 1967, and there were two publications that were critical. One was the book by Ulrich Neisser called Cognitive Psychology, which was published in 67, but the other was a paper by Saul Sternberg, which appeared in Science in 1966, in which he used this simple task, which I just described to you, and which reintroduced using response time rather than accuracy as a primary way of investigating mental processes because you could actually get more information from response time than
Yeah.
accuracy and what was actually going on in the brain. So it's a tremendous, it's the seminal study from which all subsequent cognitive experiments are derived. Now, um, well, he was right that this is the way to go. I mean, his initial experiment actually had a result which to this day hadn't been explained. And the result that hadn't been explained was that normally when you do an experiment, like in my last experiment, and you have a study string or study sequence, and then you have a single test item, there is an effect of the position when you're looking for match, the position of the test item in the study sequence, because you scan the study sequence from left to right, and therefore if the test item is at the beginning of the study sequence, you respond rapidly, but if the test item happens to be at the end of the study sequence, then you respond more slowly. And almost everyone always gets that result, which makes perfect sense. But in Sternberg's original paper, he did not get that result. He found no effect of the position of the studies of the test item in the study sequence. There was just
Mm.
no effect. Okay, and if you redo his exact experiment, you will get that result. But if you make even a small change in the experiment, then you do get an effect of where the test item is. So you have this unexplained effect. Now, it seemed to me that now that I showed there was effective response assignment, that I might be able to find out what was going on in the original experiment. And I asked, fortunately Sternberg's still around, I asked him in this original experiment, you didn't mention anything about response assignment, but did all the participants use the same key for same? And he said, no, half of them, used the right key for same, and the other used the right key for different. Okay, but he didn't
Yeah.
bother analyzing the effective response assignment in the data. So I said, okay, I'm going to redo his experiment exactly as he did it, except I'm going to do what he didn't. I'm going to include response assignment in the result. So skipping over everything else, since this is not a technical audience, I'll get to what actually happened. Um, I basically replicate his results when you do the same analysis that he does, which is that if you don't look at response assignment in the analysis, then you have no effect of the position of the test item in the study sequence. However, then I did my analysis. I included response assignment in the analysis. Now, then what happened is that I completely replicated what happened when the right key is assigned a different response, which is that there was clear search from left to right. Right? Now, what about when the right key is assigned the different response? Right? Excuse me, the same response. Now, when the right key is assigned the same response, last time I had no effect of the position of the test item in the study sequence. But this time, what I got was a reverse effect. That is, when you have to keep track of this, when the right key is assigned the different response, people
Yes.
were scanning the string from left to right. However, when the right key was assigned the same response, participants were scanning the study sequence from right to left, the exact opposite
Mm.
way. So both sides, I got an effective target position that were mirror images of each other, which is what these two figures actually show, because one on the right is plotted from right to left, and the one on the right is plotted from left to right. Um, and consequently, when you just average the two of them together, as Sternberg did, there was no effect because they
Yeah.
exactly canceled each other out. So if you go back to
Mm.
the original experiment, you can see first that you really have to include as many variables as possible in your analysis to have a good idea what's going on. And second, this is more evidence that, in fact, that more than one system is involved in this task. The initial results are misleading because the analysis hasn't been sufficiently detailed enough, but this confirms there are at least two separate parts of the brain that are using different computational procedures in order to perform even simple recognition tasks. And this has very, very broad implications for all kinds of cognitive tasks and neural health and all kinds of things. So needless to say, I have yet to get this result published, but I'll keep trying, and eventually I'll get it published someplace because I'm not inclined to give up about this. But it's
Yeah, it really
it's
looks like
important.
you put a lot of effort to not only prove it to your fellow, colleagues, but you're
Yes,
also like trying to almost like connect the dots for everybody else too. Like,
yes,
so that's really cool. And I also was thinking like, I think like diving more into these dual systems, um, or shall I say multi-system,
right.
that would be cool too. But um, it could also even help like neurological or cognitive disorders, like there's ADHD or
Yes,
autism, where there's a misfiring or a misprocessing of information that could also be, kind of dived into the
right.
systems. So I
You're
thought
right.
that was pretty cool. We are getting towards the end of our episode.
Sure.
And so usually towards the end, we do ask our own questions
Fine.
to our guest.
Go right
So
ahead.
my first question for you, Dr. Glass, is what is your favorite color?
um, it's probably blue.
Yeah. um, we should do an experiment to see how many people say blue. That'd be cool.
Yeah, probably
Um,
most. Yeah, probably most would say blue, I think. Yeah.
oh, Um, what are so if you don't have either of these, we can pick something else, but what are three movies you'd recommend and what are three books you'd recommend to our audience?
All right. Well, I would definitely recommend the movie Memento, which has a remarkably accurate description of what it's like to have anterograde amnesia. I like to
Mm.
watch movies about amnesia. Certainly more than 100 made, and most of what they show you is movie amnesia, which has nothing at all to do with the real amnesia people suffer from.
Yeah.
But this movie went to a great deal of effort not only to get what amnesia is actually like, but it's made in a particular way so you see everything from the point of view of the amnestic individual who's the central character.
Mm.
So you're suffering from anterograde amnesia throughout the movie. So it gives you this really intuitive insight into what it's like to have this
It was called
disorder.
Memento.
Yeah, yes, Memento, yeah. Yeah. So yeah, I would definitely recommend that as a movie to see. Yeah.
Mm.
Um,
Do you have any books or any other movies that you'd recommend?
Well, I could probably think of books and movies. Uh, I mean, there's so many. Well, also, there's a Netflix series which is easy, um, widely which a lot of people probably have seen already, but it's called the Queen's Gambit, and it's
Mm.
about a fictitious female chess champion in the 1950s, but it very accurately describes the mental spatial awareness and visual imagery that actual masters engage in. Chess masters can play chess in their head without the board, without the
Yeah.
pieces, and that's very accurately shown in the movie and what it's like to have this unusual skill. So I definitely and also one movie that I make my class sometimes watch is a movie that actually won the Academy Award in the 1930s, an old black and white movie called the story of Louis Pasteur, and that's what it's about. It's about the one of the two greatest biologists to ever live, Louis Pasteur, and
Pasteurized milk, right?
excuse me. Right,
He's
that's
the guy
right.
that figured out
Yeah,
pasteurized milk.
that's exactly right. Pasteur's milk, among other things. Anyhow, and more generally, the reason why I did that is he's the one who convinced the world that the germ theory of disease was correct.
Mm.
Right? I mean, in his era, there were three people who believed that germs cause disease: an Austrian by the name of Semmelweis, and an Englishman by the name of Lister, and then Pasteur. Okay, the rest of the world knew because the microscope had been around for 100 years now that there were microbes. You could look under the microscope and see them, but they thought the idea that something so tiny, infinitesimal, compared to the size of say, a human being or any animal, could possibly have any effect on them.
Mm.
So they just were not buying the germ theory of disease, and Pasteur was the one who convinced the world. And there are elements of the movie which Hollywood used to just make it a better story, but the
Right.
parts that actually deal with the science are are really accurate, okay,
Oh, cool.
and accurate to the point where like you think they're making it up, but they really didn't make it up. Like there's this tremendous experiment with sheep and anthrax. I won't give it away, but like they show the experiment as it actually happened and why it became world famous. Afterwards, all the interest in it. That's a real event that they've recreated, and not just something for movie. And the end, um, what happened when he was working on a vaccine to prevent rabies? What happened then is shown exactly as it really happened, which is that he was working on a vaccine for rabies, and he invented vaccines for anthrax already and for other things, so people had a lot of confidence with it. And rabies guaranteed you a horrible death 100% of the time. So there's a
Yep.
lot of interest in it. And before he had the point where he was ready to test the vaccine on humans, public interest got to the point that first a doctor brought him a little boy who had been bitten and said, you got to give him the vaccine because otherwise he's going to die. That whole story is true. And then
Mhm.
what happened? The Tsar of Russia told everyone in Russia who'd been bitten by a rabid wolf to go to Pasteur's lab and show up and get the vaccine. So he suddenly had, an army of people outside, clamoring.
Like free subjects, basically.
Yeah, yeah, but I mean,
Wow.
he was right. So all that stuff which they show in the movie, that's actually what happened and how he responded. So it's a movie
Wow.
well worth watching to see sometimes how science really progresses. It isn't like you make a discovery and say, "Oh yeah, that's true," but how much takes effort it takes to convince people that you're right and everyone else in the world is wrong, and how much evidence you really need, how risky the whole process can be to achieve that. Yeah, so I definitely recommend that movie also.
Nice. I noted all of them down. I definitely am growing my movie list, so thank you.
Yes.
Um, and lastly, what are your perspectives on art?
Um. Well, I mean, I'm not sure what to say. I mean, I've spent a lot of time. I mean, life going to art museums. I mean, I am interested in art. My mother was interested in art, and her sister was an artist, and so from a young age, I was exposed to art, and um, there's no question it has an important place in the world, right? And it's a place that people. I, I, I I think there's a sense sometimes that there wasn't art until there was civilization, right? That
Mhm.
like cavemen didn't have art, but then there was civilization and there was art. And I think that's probably going to turn out to be false. That cavemen and humans going back maybe nearly a million years all had a sense of what was beautiful. And for a very long time, from when they were first doing things on caves, that they were trying to create art, and they had
Yeah.
a sense of beauty and ugly, and beauty and ugliness do play a role in our understanding of the world, and so art is important in certain ways. It's
Yeah.
harder to articulate how it's important, but it clearly does play an important part in civilization, the lives of most people, whether or not you acknowledge it, but it
Yeah.
does. And then you can see that because you can see when things go when things go wrong, I mean, and things are really ugly, and they are, or things go right. Like part of it's been discussed at length. I mean, part of the success of the Eiffel Tower isn't just how tall it was, but in the end, it really is attractive. You
Yeah.
really like the way it looks. Most people like the way it looks, I mean, and that's not an accident. I mean,
Yeah.
they happen to get the right. They worried a bit over the design, and actually they went through several versions of it. The initial one wasn't attractive enough, and until they got it. So yeah, so things like that are very important in the world in terms of how much influence something is going to have. And yeah, so I do think art is something that's important, and
Yeah.
it does play an influence. If if you look for it, you can see the influence of art, and it's interesting though. I mean, different countries, different societies, uh,
they all express kind of differently,
Yeah,
right?
yeah, there are real differences. It's not the same that like it's the same everywhere. I mean, you
Yeah,
learn about different cultures by looking at their art. You see very different things and how they treat it, whether it's totalitarian, whether they have the equivalent of museums, whether they worship things. This is only partly relevant to like art, but it's like there there are many interesting cultural differences between like English and French,
yeah,
and they're particularly interesting because this societies really all came out of the same political revolution. I mean, if you look at the philosophies that led to the original French Revolution, the American Revolution, even except the English Revolution, you actually have a group of half a dozen, both English and French philosophers in discussion who imagined democracies before they're actually created, imagined individual liberty before it was granted to people, and so they have a very detailed common origin. Um, but still, their societies are different in sometimes very humorous ways. So
yeah.
some years ago,
That's yeah, that's an interesting aspect to kind of put our kind of end our show with is like
yes.
how art kind of is like the central thing that can just get bring you to the future, help you get to the past and also keep yourself stable at the present.
Oh,
Um,
absolutely. Yeah. So, I mean, by the way, if you want to, I want to give people a suggestion. If you don't know this, I mean, if you want to see art, you really do have to go to Paris. I mean, it's a great place for seeing both the most beautiful city in the world, and also for seeing fantastic examples of art. So you really want to go to Paris. But the greatest vacation place in the world to spend days or weeks really is Italy. So you really have to go
okay,
there.
guys, save up your money. We're going to Paris.
Yeah.
All
Yes,
right, Dr.
absolutely.
Glass, thank you so much for giving us your time and your energy to explain your study, what the experiment was all about, and of course, the trials and tribulations of your study going up for review and being published. Hopefully, your achievements have will help pursue other people like pursuing the same kind of dual systems or multi-system recognition of the brain. I did not know that I could understand this, so I'm very thankful that you were able to break it down for us. Audience, I hope you guys enjoyed the show as well. This episode was very enlightening for me, and I hope it was the same for you guys. I will see you all in the next episode, or maybe the other one.
Hmm.
Bye.
So long. Bye.